Are you able to see, ma'am, that I am recording?
Yes.
Okay.
Yeah. Good morning, one and all. I welcome you all for second day of the workshop on bioinformatics, molecular modeling, and biostatistics. And yesterday it was a fruitful day, and today and tomorrow I am very much confident that the same kind of the input will be given by the resource persons, and you will be able to take the same kind of a message. And today I hope that there will be some time for you to get into hands-on training, because today we have started on time. I wish each one of you an enriched learning experience. Wish you all a very good day. Thank you. Vanita, proceed to Vanita.
Okay, thank you, ma'am. So good morning to all the participants who have joined along with us for the second day on the online workshop on bioinformatics, molecular modeling, and biostatistics. So today, what we will do is that we will try to divide the session into two parts. Okay, so the first part is we will try to learn to draw some chemical compounds. Then we will be doing the auto-docking tools. By using that, we will try to do the protein ligand interaction and try to understand why are we doing this molecular modeling by using the auto-dock tools. Now coming to the first topic, that is to draw the chemical structure. Now when we want to draw the chemicals, I'll just kindly advise all the participants to have a scribbling pad or a paper or a note along with you, so that when I am telling the instructions, it becomes easy for you to follow it later. Whenever you're free, you can follow it also. Okay. Okay, coming to the first one, that is called as the Chemsketch. We are using a Chemsketch. It is a tool which is being provided by the ACD labs. ACD stands for Advanced Chemistry Development, which was developed in the year 1999. It is a tool which is used in teaching as well as in research. Now, what is the use of this particular tool? Now we use this tool for drawing certain chemical compounds. Now when you draw these chemical compounds, keep in mind that you have a previous idea about that particular compound. Are we going to use it somewhere, or are we going to use it for the teaching purpose? For the students, we have to tell to them, this is the single bond, double bond, this is the orientation, this is the two-dimensional structure, this is the three-dimensional structure, and you want to show to your students that, yes, students, this is the three-dimensional structure of a chemical compound. You can rotate it, you can see the bond angles. You want to tell it. It is very easy for us to tell in theory. As in the teaching profession, when we want to tell it, we can tell it. But when you, I am not showing any presentation, I am just discussing with you. Then I will start with my steps of how to do the ACD. Okay, so when you go to this tools, no, you can draw and you can explain to your students also. This is how it will be. So this is one of a tool which is a very free version available. We can download it and you can use it for teaching the students. Okay, so now what I will do is that I will, because we have to plan in such a way that we start with it and we can go to the docking process. Now, when I was yesterday asking if people to download certain softwares, you had some issues. It may be there, no issues, no worries. For this ACD ChemLabs, if anybody has downloaded it, okay, fine. When I show it, you can also open your window screen and you can do it properly. If you have don't have the ACD labs, I will give you other choices also. I will share few of the links with you in the chat box. You can copy that URL site, go to your Google Chrome, open a new tab, you load it there, you open the site, a small window will be appearing in which you also can practice it. The only difference is that when you are using your ACD, it has got so many features. I'll just show it to you what are they. But in the case of the other softwares which are available, the facilities are very much minimum. But for teaching the students, I feel that is also sufficient at present. Okay, so what I will do is I will go to the 1 minute. I will just try to share my desktop. 1 minute. I want to go to desktop.
আমি না একটু পরে সবজি উবজি কাটবো।
1 minute, ma'am. I am just trying to sort out.
Yeah, no hurry.
I have to take my desktop, so I am just.
Yeah, it's visible.
It's visible, ma'am.
Yeah, it's
Okay,
visible.
so the first thing is that when you are downloading the ACD Lab software, there will be 3 programs which will be loaded. One is the Chemsketch, Chembasic, and the 3D viewer. So to start with, I'm starting with the Chemsketch. You can see this icon over here, which is I placed it on my desktop. So this is the Chemsketch. So you just have to click on it, give a double click on it. The software will get opened up. If anybody has downloaded this software, you can carry on with this. You can open the software and you can start to work along with me also to understand it. Okay, so it will ask search and features. You just say it as okay, yes, I want it in this format. Okay, yes, you just click on and then you continue. It wants to give some tips. Okay, so this is what you will be having your ACD Chem Labs screen, which will be available to you. If you're having it with you, no problem, you can use it. If you are not having also, after a few minutes, I will share you the other softwares also. Now I would like to tell you about this ACD Chemsketch. What are the features available over here? This is a very interesting features which are available with this ACD Chemsketch. Now you have certain, I will just tell you what are these, all these things, and then I will draw some structures and tell to you the features. Now you can see on the top, which is given over here, which is called as ACD Chemsketch freeware. So this is going to be the title bar. Then below that you have file, edit, pages, tools, templates, options, documents, so many features, which is going to be called as the ma'am kanpistunda ma'am meku slide
Visible one.
tools screen is visible ma'am?
No, it's only desktop is visible.
Okay. Now is it visible ma'am?
No. Yeah, now it's visible.
The screen is visible?
Yes, workplace is visible.
Okay ma'am. Okay, so this is the first one. What you are able to see is called as the title bar. Below that you'll be having the menu bar, and below that you'll have so many other features present over here, which is going to be called as a general toolbars. Each one has got some important functions over here. Then you'll be having the structure toolbar, with which you can draw certain structures also. Now coming to the lower region, you can always see the cursor which I'm moving here in this spaces. You just observe the cursor where I'm taking it and going. On one side of your that is to my left hand side, you can see that there is some hydrogen, carbon, nitrogen, oxygen, fluorine, sodium. So many atoms are written over here. So if you want to draw, take any carbon, you can just click. You have to click only once on top of it and you have to take the structure from here. Okay. Then below you have some symbol like this. This is for numbering, manual numbering. You want to number, you draw a ring structure, you want to number it, then you can use this numbering tool in order to draw that one. When coming to the other end of the this particular software, you're able to see that to my right hand side, you can see that there are so many functional groups, the carboxyl group, then nitro group, then sulfur group, phosphor group. So many groups are also available. So if you want, when you're drawing the structure, you want to draw them, you can take from here also any of these and you can draw it. Now there is some add-on features also in this particular tool. Now you go to see my cursor where I'm keeping, just observe here. You're able to see here, this is called as a periodic table of elements, because you're able to see only few here. You want remaining others to be added in your structure. Now you can go to this one and you can take up the periodic tables elements also. You just click and come to your workspace. This is called as the workspace. This white color screen is visible, is it not? That is called as the workspace, where you're going to draw your structures. Now to the other right end, you have something called as a table of the radicals. So any radical you want, I just clicked it. I just gave one single click. You're able to see that so many, I just keep on histidine, the structure comes, I keep on the ring, I keep on this one. So anywhere you keep, the structure will be displayed over here. So whatever you want from here also, you can take it and you can transport it into this workspace. Okay, so now I will start with to draw the structure here, and what are the different tools which are available in this one. You can use it in order to draw it a good one, and then name the structure, and you can also find the properties of the particular compound. Whatever you are drawing, everything you can use the tools which are available in this software in order to understand it. Okay, so I will start with drawing the structure. So first thing what you can do is that, let me see the cursor where I'm keeping on the benzene ring. The moment I keep down, it is itself showing that it is benzene. So just give one small click on top of it, come back to your workspace. When I'm coming, you see the cursor, it is coming along with the structure, and just give one small click to it. Don't do double clicks and all, just give only one small click. I got the ring structure, that is benzene, in the center of my workspace. Okay, now I want to insert one more ring. And create another structure just beside it. So what you do, see how I'm moving the cursor, because if simply I'm going there, I'm just clicking, one more will come. See here, one more will come. See here, one more will come. Okay, so you have to be very clear, very much clear that when you move the cursor and when you want to draw the structures, now if you don't do it properly, then you will be getting like this rings over here. So see how I'm getting it close. Once it comes close, you just click once and I will get the structure what I want over here. So you, by seeing this, you have understood that in this workspace there are so many aromatic rings over here, but I want to study only this particular aromatic ring which I have drawn, that is nothing but naphthalene. I want to understand about this particular, but there is a hindrance over here. We have to remove all these things. So what we can do, we have an option over here to delete the extra structures which we have drawn over here. Now why we should clear the workspace and retain only with the molecule which we want to draw and study? The reason is because when you want to predict what is this molecule, what is the name of this molecule, and you want to know the properties of this particular molecule and all, no, this may become a hindrance for us. So that is why your workspace has to be very clear only with the structure which you want to further study about it. So I'm going to clear all these things. So how can I clear it that one? Now you see I am moving to one of the bars which are present on top. This is called as a title bar. The first one is called as a title bar. The second one is called as a menu bar. Third one is called as a general toolbar. There I have one feature over here which is here. It is like showing like an eraser. The moment I keep my cursor, all of you please observe the cursor where I am keeping it, so that you will know that this is the tool that is available which you can select it in order to draw or delete the structure. So just click on top of it once, come back to your workspace. See, when I'm coming back to my workspace, what are you able to see there? It is given as delete. So just click, it will delete. See, now it is deleting all the structures. You can delete whatever you have drawn the structure which you feel that you don't want it in your workspace. I don't want this, I want only my naphthalene to be there. So you can just click on the delete and all has been disappeared, and I'm retaining only this structure which I want to study further about it. Okay, now getting two ring structure is very simple for us. I have got it. Now somebody will have a doubt, ma'am, I want to add one functional group to it. From where can I do it? How can I do it? Very simple. You just see this delete cursor where I'm moving it. Go to this line which is a draw normal. Just click with that one once. Okay, come back. Now when you come back, no, so this will be having, the cursor will be having the terminal end, that is the pointer will be having a terminal end only to carbon. Now let us assume I want to insert one functional group. Let us assume it is going to be the oxygen hydroxyl group I want to assume. So take the cursor, go near the oxygen and just click with that one once. Come back to your workspace. As I told you, see here, I just keep here. Now it is what you are able to see, what has come out. But I want to insert the functional group here. Okay. So now what I do is that let us just randomly I'm drawing the structure. I'm not specifying what it is. Later we will see how to name it also. So I come back to my workspace. I want to introduce. Let me assume that this is the carbon where I want to introduce the functional group. Okay. So now if you want to put your oxygen in this particular carbon, you want to replace this carbon to oxygen. So you see the cursor, the moment the cursor I keep it on top of that carbon, it gets selected. You see a box comes near the CH, surrounding the CH. Just you click over there. Now what do you observe here? It has become O plus charges because of the in the valencies, all the satisfaction, it has attained a charge also by itself. But I don't want this oxygen here. I want OH to be attached as a extra side chain functional group. So what I can do is that you have an option here on the, this is title bar, this is the second one, which is the menu bar, this is the general toolbar. Here you can find that there is something called as undo the structure. You can just undo it. I get back to my original structure. Okay. Again, what you do is that for safer reason, you go back to the oxygen, click on that one once, come back to your workspace where you've already drawn the previous structure, go to near this carbon, don't click it, go near that carbon with the help of your mouse, hold it and draw a line. See, I'm drawing a line, you see? Yes, you are able to see, so you can see. Now you see the structure, what has appeared now. Earlier it was oxygen in the place of carbon, but now I replaced that, I have converted that one into addition of one hydroxyl group into the structure. So this is the way how you can choose any of your atom present here. If you want something new to be added also, as I told you, you have the periodic table, so you can go to the periodic table. From there you can select any of your element of your choice and you can you draw it over here and you can draw as much structure you want. But this is a free version, so I would like to tell to you that the number of heavy atoms what you take and draw using this tool should not go more than 50. So so you have to be careful with the structure when you are drawing. So drawing small molecules now, it's very easy by using the tool, this particular tool. Okay, now I want to, I've drawn, I hope everybody are able to understand. I with another short time I will share with you a small link with which you also can practice that like this only in using another tool. Okay, so now I have drawn this. I told you this is a hindrance for me. This is some water molecules which I have drawn is a hindrance for me. What is the choice? I told you the choice is I want to delete them. So go to your general toolbar, select this delete and come here and you can delete them. Okay, so it is not deleting. So I'll just delete now. You see now I have deleted this using this option, delete option. I will give one more, ma'am. What is this, ma'am? You have done so many water. Every time I have to click on top of this water to delete it. I'll give you one easy option. Now come back to your, this is your title bar, this is menu bar, this is your general toolbar, this is a structure bar. So you have here these structures, is it not? Now you see the cursor where I'm keeping on top of it. This is the fourth toolbar on which I've kept the cursor. Okay, so when I click that one, it is called as lasso on and off. So I'll go to this, just click. See, when I click it, I'll get some. Okay, I just click it, come back to my workspace and I draw something like this. Okay, so it gets selected and just go to your system. You have one option called as delete, just delete it, it will go off. Got it? So this is another option for you, lasso. If you have some big structure drawn, as one step you want to remove that structure, you have this option which is called as lasso on and off. So you can use it on and you can delete the existing structure. Another option is this one, that is the delete option, which you can delete the structure. Okay. Now I will give you, so this is how you can draw your structure. So by using the various tools which are present, the atoms which are present here, or any functional groups, rings, or some periodic table things you want, you can use these tools in order to draw your structure. So okay, so at present I've drawn a molecule, it is there on my workspace, but I want to know about certain features about this particular structure or the compound what I have drawn. So what we can do is that you go to this bar which is called as a second bar, that is a menu bar. So you have file, edit, pages, tools. Go to this tools option, just click on that one once. So a small window appears on it. You see structural properties, aromaticity, so many features are there. Let me first go to this one, that is you want to generate and you want to name the structure what you have drawn. We know the structure, we know the name, but still you want to confirm it. So you can use this option to do that one. So go to generate and you just click on the word as name for structure. I am just clicking, keeping the mouse cursor on top of that and just click once. You see what am I getting now? Earlier it was only 2 rings, I got naphthalene, but 1 OH attached, it becomes now into a new name, naphthalene 1 all. Okay, so this is how you can generate a structure for a chemical compound which you have drawn in your workspace. Okay, I hope I am clear till this. Can I have a view on the chat box? Click on the small screen displayed. Your
Maybe,
ma'am
yeah,
is
Vanita,
ah.
not many questions are there. Carry
Okay,
on.
I'll carry on.
Maybe,
Okay, ma'am.
yeah.
Okay, ma'am. So now we will go to the next one. I am not still happy with what I have drawn and I have named it. I want to understand more about this compound, what I have done here. So what I can do is that again go to your second bar, that is a menu bar, go to tools, click on that one, again a same window will appear. Now I want to, okay, I will do it undo. This is the another structure which you got it, doesn't matter. So we just go down, you can add hydrogens, that's what happened just now. So I want to understand more about the features or the properties of this particular compound what I've drawn in my workspace. So go to this word called as calculate. Now the moment I click on this calculate, what are you able to see? You can see all these features, molecular weight, formula, composition, volume, so many features you can get from this particular window property. So just you go and click on the word all properties. Now you see you get a small window appearing on top of your workspace. Now what is this? Molecular formulas, so many hydrogen, so many carbons, oxygen, its molecular weight. Just you scroll down, whatever features are there in this particular database related to this compound will be displayed in front of you. Okay, so this is how you can get the properties of this particular structure what you have drawn in the system or in this workspace. Now you can go to copy to editor. Okay, see what is happening now. When I do like this, it appears on my workspace, so that all the properties appears on my workspace. You can save this also. There is an option in this one, I will tell you. See here, go to your title bar. This is your title bar, this is the menu bar. On the menu bar, the first one is a file. Just click once, you get the option as save as. Okay, now you can save it as, just click once. Now it is telling, array, you have drawn the diagram, you should guide me, where should I save it? Okay, so this is how it will come. So you can go to desktop, wherever you want to save your structure, in that particular place you can save it. So you can create a new folder and you can save it. So when you're saving it, please give the file name. As you now drawn the structure, you've got the properties, you've got the name of the molecule. So better you can name the compound and you can save it. But one kind information to everyone, please make a note that when you are saving this, the file type in which you're saving is very, very important. Okay. Now I'll just click on top of it. Again, small window appears telling in what format do you want me to save the structure. Okay, so now there are so many formats available in which you can save it. This is mol file format, you have IS sketch format, MDL, RxNile, so many formats. FASTA format is there, paintbrush is there, JPEG format is there. But for future studies, I will tell you where this ACD Labs is going to help you out in the next step. So when you are saving the structure, most of the people, they go only saving it in a format which is called as the mol file format, which is a very common format in saving the structures, which will be helpful for us when we are going for research or when we are going to use this molecule for the docking process. Okay, so just click on this mol file format and you can give the word as save. So it will get saved in your document. Okay, now I'm not saving it, but this is the step what we generally do it in order to save the molecule. Okay, so now you understood, you have drawn the structure, you have named the structure, you've got the properties of the structure, whatnot. You're able to get so many features about it. I will add some more features about this structure what we have drawn over here. Now I got this one structure. I want to go with the 3-dimensional structure optimization. So I just click on that one saying, okay, select. I will try to remove this one. Okay, so I'm having on my screen with this structure, which is naphthalene. 1 minute. I hope all the participants are able to see and learn something today. So let us make it that we learn something here. Okay, so this is the naphthalene structure which I've drawn. Now you can see some features over here on my screen. To my right, see my cursor to my right, what I am showing over here. This is an option over here called as a smiles notation. So you can name like this, or you can also get it as smiles notation. That is another way of writing the structure. Expansion for smiles is a simplified molecular input line entry specification. So I just click on top of it. Just to click on top of it, see what is happening on the screen. So something has happened. Let me make it very clear. 1 minute. Okay, now I'll go back to the smiles. So just to see what has happened. Now this is explaining to you about the, see this is O. So this is the oxygen atom. Then this oxygen is attached to the carbon. So this is considered as your first ring. Okay, so this is the carbon, then second carbon, third, like that, and it is coming and going into the second one. So as I remove, the smiles notation is also changing. Okay, so I will just do it as undo. Got it. So this is one way of understanding the way of representing the arrangement of atoms in the molecule what we have drawn over here. There are so many other features also. I will give one interesting feature to everyone. Once you have drawn the structure, no, you want to view it in the 3-dimensional viewers. Okay, so let me show you this feature also, how you can do that one. So this is an add-on feature which comes along with the ACD software tool, which is not available in the other software tools. Okay, now you see the structure how it has appeared. Earlier it was different, now it is, you see, it has come in a 3D viewer structure. Okay, so I hope all of you know what are all these things. These are the ball and stick model. I am not happy with ball and stick, you want to convert it, no problem, just click that one, it comes into wireframe structure. So like this, you have so many options on the screen. You can see on the top you have so many options. By using that you can change your pattern. Okay, I am retaining this ball and stick model. Okay, I'm still not happy. I want to show you something very interesting with this one. Okay, so what I can do is that I go with this one. This is called as atoms color setting. Okay, just click that one, a small window appears. As such, background is black. I'm very happy with the black color. Now I want to make sure that the oxygen which is there on my screen, I want to give some bright color for it. You want to make it very interesting. So just click it. Okay, now the color has changed, you see. So you have an option.
The screen is frozen. We cannot see the 3D models.
Ma'am, Sai Padma ma'am, 3D model is not visible.
Yeah, it's only line drawing is there.
Okay.
Ball and, yeah, you have said ball and stick model, which has not come.
Your screen sharing is paused is coming for me as an option, ma'am. I don't know. I'll just stop it and come back again. Thank you, ma'am, for telling me
Yes.
what has happened. I'll do again, ma'am. Now is it visible, ma'am?
Yes,
Okay.
Manita.
Okay,
It's visible.
okay. Thank you and sorry for the inconvenience. I think you are enjoying it. Okay, thank you. So this is the atom and you like this. This is carbon. You want to color it differently. You have this option.
Even I'm just thinking it's going to come. But before that only somebody has raised.
Okay,
nice that they are very keen in observing.
Okay, ma'am. Thank you, ma'am. Okay,
Yeah,
now I'll show you something also on the menu bar. You can see this option is there. I've just put the cursor on top of it. Just click once. You see what is happening. I didn't do anything. It is auto rotation mode. I kindly request
See,
the
Vanita,
participants
a
to
small
mute.
interruption. Somebody is asking how you got this ball and stick. Maybe just one step behind you, please go and then explain.
Sure.
Because in between the screen got frozen.
Sure.
You please explain.
I will just wait for its rotation. See, I have stopped this. This is ball and stick. My dear participants, please mute yourself. I'll be very happy. You can rotate it even with your mouse also. This is auto rotate option. What I've done, see on this bar you have so many tools, is it not? So this is the first one, that is a wireframe. Are you able to observe now, all of you? So the structure will change to wireframe. And you see one more thing, when I keep it near these atoms, you see the XYZ coordinates are also coming. So you get lot of information with this ACD tools. Okay. So next one is sticks. Only sticks will come. Okay, then this is, are you able to see now? So this is ball and stick option. Just click once, the structure by itself will get converted to the ball and stick model. I'm just moving it with my finger on my laptop with my finger. I'm just moving it and rotating it. In what angle you want to rotate, see the structure you want to, you can do that one. Otherwise you can go for this auto rotate option also. You have space filling option also. You have the dotted option also. So it's up to you how you want to view the structure. But preferred one in chemistry, generally they go with the ball and stick model. So you can use this one. So you feel that this software is so nice that when you show these sort of structures to our students, who will not love chemistry? Every student will start to love chemistry and becomes very interesting for them. Okay. So there are so many other features. You can as well experiment at any time once you have downloaded this ACD lab tools. It's a free software only, no issues. You can. Okay, again I have to share my screen. Okay, now it is visible, ma'am. So this is we are back to our own original workspace where we have drawn the structure. Okay, so you have I hope you have got this how you can use this tool in order to draw it. I'll give you few more add-on options also. I'll just tell the features. You see you have here certain bonds which are present. You want to convert some bond into like this, like I just click that one, come back here, you can insert it. I'm sorry, it is not inserting. Undo, come back here, let me go with this. You can insert these bonds also. Okay, I will show it to you with another format also, how you can do that one. I'll go back to this carbon. I'll just click on it once. Okay, and I come back to my one, and you see the screen. I am just drawing it. Are you able to see now the structure? You see this bond. You want to, in chemistry, these bonds all play a very important role, is it not? So you can use these stereo bonds in order to draw the structure and explain the importance to our students also. Okay, so I am just erasing it off at present so that I will show you something else also. Now you see this tool now, it is this is free software. There is also a commercial one which many people are buying it. The reason is because once you make it as a commercial option of taking this software tool, no, you have one option where the diagram whatever whatever structure you're drawing, no, you can make that structure. You can use it for even your paper publication purposes also. So this tool, drawing the structure, is being accepted in few of the journals, because in chemistry, especially in one journal I read, that it is Journal of Biological Chemistry, they are telling that the structure has to be drawn, presented only in this particular way. So you have to prepare your structure in such a way that it becomes easy for the publishers also to accept your paper. So this is one of a tool which we can use, and you can draw it and you can use it for publication. So since it is a freeware, I'm not able to show it to you, but this is how you can go and see it. So let's go to help. You have this reference manual. So in that reference manual, they have the supporting system. Since it is a free one, it is not showing me the option, but if you are going to purchase it, you will be getting this option. You can take it. Okay, so these are the few things which I would like to tell. There are few more. There are a lot of applications with this particular tool. So you have here something called as templates. Okay, so I'll just show you this one more feature and then move on to the next part of our workshop. Okay, so templates. So you go to this template window, just click it on that one window. So you see so many structures are already previously been deposited in this KCD Labs menu bar. So you can use it. Now what I can do is that, you see, this is alkaloids, you have aromatic compounds, you want to transport this compound structure to your workspace, you can select them and you can transport them. Then you have carbohydrates, Fisher projection formulas in the form of open ring structures, carotenoids, DNA, RNA kit, HPLC equipments are there, because I told you that sometimes you can use these tools, this particular structure what you're drawing for your papers also. So you can, if you want to draw it, you can use lab kits, because in chemistry these are the very important components in the chemistry. Okay, you want to explain to your students every day, we cannot carry the apparatus, but you have this software tool, you can download it in your system, you can take your students one day, show them all these things, they'll be very much excited to see it. Then you have phosphorus compounds, steroid compounds, sugars, terpenes. So all these compounds are already available, so you need not draw these things. If already they are there, you can just transport it from this region into your workspace and you can use it. Okay. Okay, I hope you are excited with this tool. Can I see the chat box? What is your opinion about this one? So you have understood? Okay, I am crossing my time, 10:41 it is, so let us go with the next tool. So you can, there are so many features. You have reaction, you write one compound, you put the plus symbol, add another compound, you can use this tool, you can put, I'll just show it to you. So that's, see, I have drawn one reaction, forward reaction, then you can go to this one arrow, labeling, you want to label some conditions, some parameters you want to include, so you can go and so you can go to this small window will appear. So top you have one, because above the arrow mark you want to mention something. Let us assume I want to put this symbol, and down you want to indicate what temperature is the heating temperature. You can just put okay, it appears on top of your reaction. So there are so many features available. What I told you is very much minimum. You want to explore it, you can just download it and you can experiment it. Okay, so it takes 10 minutes for downloading this particular software, but still you can try it out. Okay, so I am done with this one. Now question comes to you. Why ma'am did she talk about, why did she talk about this Chemsketch, and then she is going on to talk about the next aspect, that is molecular modeling. The thing is that when you are going to do this molecular modeling, one of the strategy is protein ligand interaction. Now you have a protein, you have a ligand. Okay, so you can download the ligand structure from any of, I'll give you the databases, two databases which I have been referring, I'll tell you. So use that one and you can take the ligand structure. But suddenly you feel that that is not the structure, that is not the ligand which you want to work with it, and you're not able to get the data of that particular ligand. But we cannot stop our work, is it not? So by using this tool,
no, we are not supplying women milk currently. in 90 and it's very difficult to cut movement.
My participants, please mute yourself. Please mute yourself. Okay, so what we can do is that by using this tool, you can draw the structure. You can draw the ligand, provided, as I told you, molecular structure must be small. Maybe if you are making it as a commercial one purchase, we may have the eligibility draw it. But generally, ligands will be less molecular weight compounds with less than 500. If you want to use it as a drug, as per Lipinski's rule, its molecular weight must be less than 500 daltons. So small molecules you can draw here, and as I told you earlier, the file format, extension file format where you are going to save this particular structure will be in the mole file format. Remember the format names I'm telling to you. You can note it down in your scribbling pad also. So by using this tool, you are drawing the structure and you are saving it as in what format? Mole file format. Okay. Now that is still not sufficient when you want to do the docking process. So you have to convert this particular mole file format into a very important format which is called as the PDB format. So whatever structure you're drawing over here has to be converted from the mole file format to the PDB format, protein data bank format. So that is a format which has been recognized when we are performing the docking process. So that is the advantage of this particular ACDs tool. You can use it for education purpose, you can use it for research purpose, but when you are using it for this ligand drawing and using it for docking purpose, then make sure that you convert the mole file into the PDB file. Again, we'll require one more important software to convert the mole file into the PDB file format. So that is called as OpenBabel. One of the software is OpenBabel software, with which you can convert the mole file format into the PDB format. So remember that when you're starting with your docking process, you require a ligand. So the ligand, what you can take, it can be
Yeah, Vanita, carry on.
so when we are drawing these structures, yes, PubChem is also one. I'll give you the certain tools also where you can take up the ligands. Okay. So if the ligand is not available in the PDB format, then you can come back to this particular tool. You can draw it, convert from mole file into the PDB file, and then you can use it for your experiments or for docking purpose. Okay. So I'm just closing this window and just will go on because time is very short, so we have to go with the next important process. Okay, I'm just closing this one. So it is asking me, do you want to save? At present I'm just giving no. Okay, now again what I will do is that I will come back to my
Ma'am, sorry to interrupt.
Yeah.
Ma'am, can we have a small question answer, sir? Because the doubts which we are having in this session, can we ask now if possible?
I am very happy to take up the doubts, ma. But can we have it later?
Vanita, maybe one or two questions you may just give.
Okay, ma'am. Okay, ma'am.
Them.
Okay, ma'am.
Not
Okay, ma'am.
many questions if they are very much relevant and.
Yes, ma'am. Because ma'am, we are directly saving in the mol format. Ma'am, can't we save in a PDF or JPG and use it in our presentation?
You can use it for which one? If you want to use it for your students to teach it, you can use it. But that format, you cannot use it for your docking process.
Okay.
Because, yes, ma'am.
Yes, ma'am. Next question is that, ma'am, suppose in the 3D structure, structure, we are able to differentiate between the double bonds and the single bonds.
Okay.
But in the 3D structure, we are not able to differentiate between the double bond and the single bond.
Yeah. That is because of the diagrams in which it has been represented. In that case, you can go, instead of going with ball and stick model, go with your wireframe model. In that, you are able to see the double bonds. Got the answer? You have options. You have options there. You can. That is what is the advantage of these tools. You have the structure in the 3D form structure viewer only. You have wireframe. You have the ball and stick. You have the line model. So in the wireframe model, you are able to see the double bonds there. Is it not? So you can use that option over there to understand the position of your double bonds. Got it?
Ma'am, what if we have any novel compound which has been identified recently? So can we carry the sketch of novel compound with this platform?
Yes, you can carry it, sir, no problem. But as I told you, it will have its own limitations also. What is the size of your molecule? Okay, so as for students' point of view, you can draw the structure and you can tell to them the arrangement of atoms, their location, 3D, you can explain to them. Okay, but is that structure, can you convert it into the PDB format, and when you can use it for your docking, you have to be very careful with that one, because when you want to use this particular compound as a ligand for a docking process, it should follow one important rule, because the ligand what you are going to use for your docking process, we are going to consider later maybe as a drug. Okay, so if you want to label this ligand to be as a drug after optimization, hit identification as a potential drug, it has to follow the Lipinski's rule. First important point of the Lipinski's rule is what? The molecular weight of the compound. How much it must be? It must be less than 500 Daltons. Okay, so that is why I'm telling to you. So we can draw the structure. This is for teaching purpose. We can explain to our students. Okay, but when you want to do it for the Autodock purpose, then you have to finalize the ligand what I'm drawing. Can I use it for my next step or not? Will it be utilized for that step or not? You have to finalize all these criteria, and then you can use this tool in order to draw them. Okay.
Yeah, Vanita, you can carry on.
Okay, ma'am.
Any questions they will post in the chat box, because
Okay,
otherwise the time will become a constraint and
okay.
the resource person is there.
Okay,
Otherwise, at any point of time, they can mail you.
yes, ma'am. Sure, ma'am. Anytime I will be answering them for any questions, any help. I'm always there with you participants. No worries. Okay, now we'll move on. Now we'll move on to the
answer session at the end of your session for 5 to 10 minutes, and
okay,
we're noting down all the questions. That time you can answer.
okay, ma'am. Sure, ma'am. Okay, so now we are talking about, we'll pass on to the next step of the today's workshop, that is molecular modeling. So it just is brief introduction about it. So it involves theoretical methods and computational techniques to model or mimic the behavior of molecules. It involves certain features where the important point over here is that atomic level description of the molecular system is what we are going to understand over here. The goal is to develop sufficient and accurate model systems to avoid physical experiment later. Now, by using this molecular modeling methods, we're able to investigate the structure, thermodynamic, surface properties of biological, inorganic, and polymeric systems. Now, what are the, when you go to the depth of this molecular modeling, it has been used in research, and we are able to understand the biological activities like enzyme catalysis, protein folding, protein stability, conformational changes, molecular recognition of protein, DNA, membrane complexes, etc., and many other features also. Now, coming to this molecular modeling, you have certain options in your hand, like you have a receptor in your hand. Okay, so receptor is a macromolecule. It can be a protein, or it can be an enzyme also. So you have some receptors. Many receptors are there with you in your hand, and you are so unfortunate that for some of the proteins or the macromolecule, you don't know the structure of it. Okay, fine, but you have certain other receptors for which you have know the structure of it also. Okay, you have one more option in your hand, that is called as a ligand. Okay, so there are certain, these are the, I'm trying to tell you about the strategies what you can follow for molecular modeling with the available sources in our hand. Okay, so one source which is available with us is the receptor, the other one is the ligand molecule. So you have ligand, you have certain ligands for which we don't know the structure, but for certain ligand you know the structure about it, or the physical properties, all the properties you know about it. Now you have two options. You have a receptor, you have the ligand, you want to study both of these together. So what strategy you would like to follow now? So there are four options for you. The first option is called as a homology modeling, what yesterday ma'am was also talking about it. Okay, so you can see that you have the receptor, but the receptor what you want to study, you are not being provided with a three-dimensional structure of it. Similarly, you have a ligand, but for that that also you don't know the structure about it. So what you can do is that first you try to build the lock. You consider this to be as a lock and try to fit any of the key which will fit into this lock. Okay, so how to generate this 3D structure? By homology modeling you can perform it. Okay, what is the second option? Now this is the receptor macromolecule. You have the structure with it, but so unfortunate that the ligand, you don't know the structure of a ligand. With which ligand you want to work, you don't know. So what you can do, build or find the key for it that will be able to fit the lock. That is called as a receptor based drug design, or you call it as de novo drug design. The another one is you have a bunch of receptors, but you are not having the three-dimensional structure of it, but you have certain ligands and you know about the structure of it. So you can go with ligand based drug design or indirect drug design, where you will be performing two-dimensional or three-dimensional QSAR, quantitative structure activity relationship, and you can find the infer the lock by expecting the key. The last one is you have the receptor, you know the structure of the receptor, that is a protein or a macromolecule. You have a ligand, you know its structure, properties, everything, and then we can try with this strategy which is called as the rational drug design, or it is structure based drug design, where we are going to talk about this one only today. That is called as a molecular docking, where you can take this drug, interact it with the receptor and try to understand about it. So there are 4 different strategies available based on the various parameters in the case of a receptor or a ligand, whatever is available to us. Okay, so now for performing this molecular docking, one of the tool which is available is called as a Autodock. So this is actually a software which is freely available. There are different versions, 3, 4 and 4.2. 3 and 4 are old versions, 4.2 is the latest version. So if you want to follow the Autodock tools procedure to perform this molecular docking, so what is molecular docking? You have a protein in your hand, it's a receptor, it can be enzyme, it can be a huge macromolecule. You have it and you know the structure also of that one. Then you have the ligand, you have a ligand which it is a key which can fit into the macromolecule also. So if you have that one, then you can do the molecular docking, where you are trying to make an interaction between the protein and the ligand to understand the protein and the ligand interactions. Okay, so these are the steps what we are going to follow. And why did I take this tool, that is a Autodock tools? Because it is widely used in literature. It is the most cited docking software. It is very fast and it gives high quality prediction of ligand conformation, and you can perform blind docking. So this is very important, because whenever we take a protein, when the data, when the three-dimensional structure of the protein is available, sometimes you may not be able to locate the active site of the enzyme molecule. So that time we go with the docking called as a blind docking, and it is a free software tool which is available for the general use. So that's the reason why Autodock tools is very famous for molecular docking or to understand protein ligand interaction. Okay, so with this I will just go with my next one. I'm going to talk to you about Autodock tools, how to do this one. 1 minute, ma'am. I'll just share my screen again. I have to minimize. I'll just close this. I'll share my screen again. Okay, so this is the screen. I'm just sharing with you. Are you able to see the screen, ma'am? Autodock tools, I've opened it up.
Yeah, it is visible, Vanita.
Okay, ma'am, I'll carry on now. So this is the Autodock tool. See, to my right hand side, this is my desktop. You see there are so many. Yesterday, this is the software which I shared with you, but many of you were not able to download it. Not at all a problem. At any time later, you can try it out with those softwares. So this is the tool which I have just logged in. That is the Autodock tool. This blue color icon symbol is there. Just I have logged in. So I just put it as remind me later. And this is how our screen for the Autodock will appear. Okay. So I'm just maximizing it. I hope the screen is visible to everyone. So I'll first explain to you about the features of this Autodock screens, and then I will go with the steps of how to perform the Autodock. Okay. So you can see there are so many features available on this screen. So you have the first one, which is called as the title bar. So that title tells you Autodock tools. So that is called as a title bar. Then you have the next bar, which is called as a menu bar, having file, 3D graphics, edit, select. So if you go to edit, click, you will get so much list of points listed out. We will come to each one of the properties later. So this is going to be one of the toolbars. Then this is called the second general toolbar, which is available. And this is the bar which is we are going to use it for the auto docking process. So we have ligand, flexible residues, grid, docking, run, and analysis. So many features are available over here. Okay, so these are the things which we are going to use it for our docking process. Next, coming to the lower section, you're able to see this is the lower part of it where I'm moving my cursor. You're able to see blue color lines crossing here and there. So you come to the left end of it, you can see all molecules, current selection. Since I've not selected any of the molecule, there is no chance of any structure visible here. Okay, when I select the compound, you will be able to see here. That also I will show it to you. Then you have certain features over here, like you call LB, that is nothing but line formats, ball and stick models. You have the ribbon model, or you can color, you can give a color to the compound, whatever we are going to display it. Okay, so now in the center, you're able to see a screen which is black in color. So this is called as GUI, graphical user interface. And this is the place where you're going to have your ligands and the molecule, macromolecule, which we are going to use it for the docking purpose. Okay, so before starting it, I will give you two important points with it. Okay, the first point, remember that this is protein ligand interaction. So you're going to take a ligand and you're going to make it to interact with the protein molecule. So you have to know the structure of your ligand. You should know the properties of the ligand, and then only you can check out whether it will be fitting with your macromolecule, and you have to use it. So there are two ways of taking the ligand. One ligand where you can take it is called as the ACD Chemsketch. What I told you, if you are able to draw them, you can convert to mol file, then convert it into the PDB data bank file, and PDB file is only format which will be recognized for the auto docking. So structures has to be downloaded, taken only in the PDB format. Okay, so this is one way you can do it. Or if you can, that is one format you can do it. The other way is, I was searching for certain drug database centers, and I found there are 2 or 3 drug database centers which I came across. I'll share it with you, the names. If you have a scribbling pad, just make a note of it. One is called as a drug data bank center. So from there you can retrieve your drug molecule for your study directly in the PDB format. That is the significance of the database center. In ACD, you have to store it in the mol file, convert it into the PDB. But these database centers provides a structure directly in the PDB format. So directly you can download it from the data bank center. The second one is the zinc database center. When I was trying to understand, it is telling for commercial purpose, but still there may be some small molecules available as a ligand for us to study. So you can download it. There also you have to take it only in the PDB format. Okay, similarly there is PubChem also. From there also you can download the structure of the ligand. Please remember that. Don't think that if I have this is a macromolecule, this is a ligand, what I would like to dock with it and study it, and go in search of these ligand names in these database centers. What I told you, sometimes you may get it, sometimes you may not get it also, because the database centers have their own limitations to circulate some of the free versions of these drug to us. So all may not be available, and also all ligands or all drug compounds may not be deposited even in those database centers. So if it is available, well and fine, you can start with your work. If it is not available, then you have to draw it, convert it from the Chemsketch, that is a mol file format, into the PDB format, and then you have to use it. I hope you are sure you are clear with this point. The second component what we require for auto docking is called as a protein macromolecule. And this protein macromolecule, yesterday we were seeing about so many databases database centers. You have GenBank, NCBI, all are nucleotide database centers. But one of the well-known database center is called as the RCSB PDB Database Center. So PDB is for Protein Data Bank. So by using that database centers, directly you can retrieve your macromolecule protein of your study even from that database center. But again, important point, please remember that you have to download it only in the PDB format, Protein Data Bank format. What is this format? This format is very simple. It is like a text document, and it will be, as you go, when you open that file, when you download it and open the file now, you will be having all the atoms and what are its coordinates. All those things will be displayed in the form of a text one. That's it. But the data which is available inside that plain text format is completely in the PDB format, which can be recognized by these Autodock tools. So that's the reason why I'm continuously specifically saying that when you want to do the Autodock, make sure that the ligand what is available to you, the protein what is available to you, both of them are only in the PDB formats. Okay, so I've given you the locations where you can take all these tools. Later I will try to share with you all the sites so that you can experiment later also. Okay, so now for the today's purpose I've taken ma'am. Ma'am.
You have 1 hour time. Maybe
Okay.
manage within that 1 hour.
Okay, ma'am. Should I ask them to, if they have downloaded the program, I don't know whether they will be able to run it, because I have to show them, means it will take time. Can I run the program, ma'am? Autodock program?
Yes, you need to know, but you just see that you will finish little quicker, and then.
Okay, ma'am. Okay, ma'am. Okay, ma'am. Okay, ma'am.
Those who have downloaded, they will be able to follow.
Okay, ma'am.
They will follow later, because
Okay.
anyway we are providing the manual.
Yes,
so
ma'am. E-manual is
a
there.
little faster pace only, so
Okay,
that
ma'am.
you will finish and Mr. Ramesh will join.
Okay, ma'am. Okay, ma'am. Okay, so as I told you earlier that we have certain steps in performing the Autodock process. One is preparing the ligand, then preparing the protein, setting up the grid, then grid parameter setting. You should do docking parameter, auto grid, auto dock, and then you have to generate the results. Yeah, I know that it is all like Latin and Greek for you. Not at all a worry. So you have to practice it. As more number of times you practice it, what I'm telling you the steps, it will come very easily for you, and you can just do these things very easily. Okay, so as I told you earlier, I'm going to start to do the Autodock process. I'm going to take a ligand, I'm going to take a protein molecule, dock it, and within 1 hour, I'm going to see probably that I can show you till the last that how the ligand, what I've taken, is able to dock inside the active site of the protein molecule. Okay, so now you should understand that when you take a ligand, when you take a protein molecule, you're trying to put them together, you're allowing them to interact with each other. So at atomic level, what atoms of the ligand is interacting with the macromolecule, that is what is very important in this docking process, because the atoms which are interacting with the macromolecule, there will be some energy released out, and that energy what is released out must be minimum energy. That energy is what is called as a binding energy, what is very, very essential for us to understand how this ligand with a particular pose or a particular conformation, its atom is able to interact with the macromolecule. So that understanding is very important. This is what I'm going to show is only execution of the program. Okay, but interpretation of the result is very important. That is the last step. So I will try my maximum level best that I show you till the last, so that you will enjoy exactly how this tool is working. Okay, so what I've done is already I have downloaded the PDB file format of a ligand, and for today's ligand I've taken this imatinib. I was able to generate, I was able to download the structure from the drug data bank, so you can also try that one. And the macromolecule what I've taken is 4-MXO. It is a human SRC kinase protein enzyme molecule. So these two I've downloaded directly from their respective sites in the PDB format. Okay, so now I'm going to tell you how to start with your docking process. Okay, Now I told you this is toolbar, this is menu bar, this is general toolbar, this is the command, the bars which is available over here. See my cursor where I'm putting it, I'm just putting it on the ligand. Okay, so now before going to this Autodock process, step number 1 is that you have to have a very clear idea that where have you saved your PDB formats of your protein and the ligand in your system. That is very important, because continuously the program will ask you the location where all these programs are being kept inside, so that the docking and the running process runs very smoothly. So first of all, what we will do is that we will set certain parameters over here. So you have the first one called as file. Just click the file, a small window appears like read molecule, read session, save, so many options comes, but I'm preferring to take preferences. So go to file, go to preferences and click the word as set. The moment I click that one, a small or a big window appears over here, like asking you so many data, but I'm more concerned about this criteria. Just observe my cursor and listen to what I'm telling to you, to participants. So kind of you listening to me so patiently. So this is the I can show you this here. This is called a startup directory. So there is something written already here, so I don't want this. I want to tell to my Autodock tools that this is the place where all my data are being kept at present. So you have to take it from there. So what was previously existing, I just removed it, I deleted it, and you just go back to your system, and I'm taking my folder. I hope you are able to see my folder. So what I did is, on my desktop I kept one folder as workshop 13, and I kept 3 different documents over here, like Autodock, documents and video, and I'm just clicking on this Autodock. So now we are able to see that there are so many features available over here, which is 1 minute. I will just go back. I'll just, ma'am, screen, you are able to see the screen, ma'am?
Yes, Vanita, we are able to.
Okay, ma'am. So I'll go with this format where I'm having all the data previously. So I will just go to this one and this one. We will generate the data from here. So now you can see that I have on my desktop, this is the place where I have saved all my documents for today's auto docking purpose. Now 4MXO is a protein, that is a macromolecule which we are going to use it for our today docking purpose. Now you see something else over here called as DB00619. I told that ligand or the drug what we're going to use is called as a imatinib. Okay, so now instead of imatinib, the drug database has defined some numbers for it with some accession number. So this is the number of that particular compound. So instead of imatinib, I'm just continuing with the same format. So as I told you, from the drug database, I've downloaded this particular structure. Please see to this type of the file in which I've downloaded, PDB file format. So this is very important. Similarly, the protein what I've downloaded is also in the PDB format. So now I understood where my files are there. Okay, so now what you do is, on the top you have program to share it. Just give a right click to it and you say copy the address. That's it, and minimize it. Come back to your startup directory and you give telling that this is the place where my documents are there. Please take it up from there to follow the Autodock process. So just put the word as set and then you can close this one. So you can just close this. Okay, so the moment I close, I have already fed the information. This is a place where my documents are there. Okay, so participants, please, I'm going to start with the docking process because I already have the data with me. I'm just doing it. So don't worry. Anytime you can approach us, we will always guide you how to do this process. Okay, so it's a little bit time-consuming process. We want you to enjoy and experience it till the last. That's why I'm just speeding up the process. You first enjoy it, learn it, then you can practice it at any time and we can help you out. Okay, so step number 1, we are going to prepare the ligand. So as I told you, have this file graphics and all these menu bars. I just go to ligand, see a small window appears, go to input and just say it as open. Now you see a folder has opened up. Okay, remember what I told you, I have saved my documents in one file, and now I'm giving it, this is ligand, please open it up, but it is opening and my space is empty. This always appears. Please remember, don't panic when you are doing the process. It will be always empty. The reason is because, come down, you're able to see that files type. What is a file type over here? PDBQT. But I didn't tell you to save it as PDBQT. What is the format I told you we have to save it as? PDB format. The moment I select PDB format, you see what appears on my share it folder. You are able to see 4MXO, that is a protein or a macromolecule, and this is the ligand. Now you click on top of that one and you just open it up. You see something comes on top of your GUI. Okay, so this is the ligand imatinib, which we are, that is nothing but DB00619, which appears on your screen. Okay, so that contains all the features about this particular ligand appears over here. Just say it as okay. Okay, so step number 1 is preparing the ligand. So go to the ligand bar and click it. You'll get input, open it up. You have to open your, it will open the folder. Just select the drug name and the drug appears on your screen, and this is the drug structure. Okay, now you come back to your screen. Here you see I'm moving the cursor here. You are able to see that current selection. You have the drug compound DB00619. Okay, so earlier it was empty. Now you have got the structure over here. So this is the structure what has appeared. Okay. Now what is the next step? You have to save the structure. So go to ligand, go to output. It will ask in what format you want. What I've done in this stage is that I've prepared my ligand, converting it from PDB format into a next format called as a PDBQT format. What is this PDBQT format? So repeatedly I'm telling PDB, that is protein data bank, and Q stands for charge and T stands for the type of the atom. I told you molecular docking or molecular modeling, we are studying at the level of atomic level structures. Atomic level structure, we are studying it, so we have to save this one. So you just click the word as save as PDBQT format. So now what you can do, the file is actually, the folder is empty, no worries. You just see down DB00619, that is a ligand. Just put dot and type it as PDBQT format. So this is the format where the further programs will recognize the ligand molecule. So it is very, very important. So remember, you started with the ligand in a PDB format, you have converted it into a PDBQT format. So when you give the first step of your program, you're telling that this is the charge, what it should be attained. Everything is being done by the program itself. Okay, so we have converted PDB into the PDBQT format, and you have saved your ligand. Coming to the next step, you're going to prepare your macromolecule or the protein for the docking purpose. Now I finished with ligand, I have to do with my macromolecule. So go to the file and just click the word as read the molecule. So the moment you click, directly the directory shows you where is the compound deposited. So already you know it is 4MXO, that is a macromolecule. Just click on top of it and say it as open. Now wait for some surprise. You are able to see on the screen, you are able to see this is the protein of our study, what we have taken as a macromolecule. Okay, so you can make this one as I told you, this is 4MXO. You can see here, this is red color thing which is being displayed over there, which is in the lines, and this is the ball and stick model. This is going to be the atomic sphere model. This is the ribbon model. So if you want, you can click it as ribbon model. So now you can see that you are able to see the protein in the ribbon model pattern. So you can just, with my mouse, I'm just rotating it to make you a view of the whole structure. You can see all the alpha helices, beta sheets, all are beautifully been appearing it on top of our screen. Okay, now I'll go on to add few more points about this particular protein molecule. Now coming to this protein molecule, I just see to my left hand side, what you are able to see is that the protein, you have the ligand, you have already placed it on your GUI. The second one is a protein molecule 4MXO. So just click on top of it. So you can see something else appearing on your screen. There are two chains. You see the symbol over here, it is showing you that the protein what I've taken is made up of two chains. One is called as the A chain, the other one is called as the B chain. Now, when I'm taking this protein molecule, you have to remember that when I'm taking this 4-MXO protein molecule from the protein data bank, I have downloaded it in what format? PDB format. Okay, so by using X-ray crystallography or by NMR studies, they have crystallized the protein molecule with already an inhibitor, and they will be depositing in these database centers. Now, why did they put already an inhibitor and they are going to save these structures or study it in the X-ray diffractions? Because to make the binding site of the protein molecule in a stable conformation, so that anybody is taking that particular molecule from the database center to study about it, they can remove that already pre-existing inhibitor, and now you can put your molecule of interest into that. So your structure, that structure what you're taking now, it is already in a stable conformation. So it becomes easy for you to understand the interaction of your new ligand with the active site of the enzyme molecule. Okay, so that is one point. The second point is that proteins may be a single polypeptide protein, or it may be a dimeric protein, or in different you know that protein about proteins. Okay, the protein what we have taken 4-MXO is going to be a dimeric protein made up of A chain and the B chain. So for today's experimental purpose, let us go only with the A chain. We will try to remove the B chain because the A chain is having the active site. So I would like to do my study, or I would like to show my session by taking only the A site. But please be very careful when you're taking the protein. You always have to take the protein as such. When you're going to do the docking process, first to finalize where the active site of the protein molecule is present, whether it is in the A site, whether it is in the B site, everything you have to have a clear idea about the protein on which you're going to work with it. So based on that, you can finalize which chain I want to retain and which chain I want to remove it. Okay, so in this study, I am trying to remove the B chain and retain only the A chain for the docking purpose. Okay, so now how to remove the B chain, I will tell it to you. But there are certain proteins where sometimes the active site may be spilling over A chain and the B chain. In that case, you cannot remove the chain. Okay, so that is the reason I'm telling you that when you are taking a macromolecule for a study purpose, first you understand the complete structure of all the components which are present in that protein molecule, its conformations, active site. Do you have the data about the active site? Yes, I have. No, I have. Then I will tell what to do with that one. So you have to finalize with all the relevant information available related to the protein molecule. Then only you can experiment yourself with the docking tools. Okay, so now coming to this B chain, I want to remove this B chain. I want to retain only A chain, I told you. So you see the cursor, what I'm doing, I'm just, you have the B chain coming straight to it. I have one small round over here. Just click on top of it. The moment I click on top of it, see what has happened over here on the GUI. The chain has been selected now for the deleting purpose. So I just selected, and now what I do is that now I'm going to remove it. See the movement of my cursor. So you just go on top onto your file, a 3D graphics, and edit. Just click the word as edit, and you just delete here as delete the selected atoms. So I want to delete the B chain. I want to retain only the A chain. So you select the B chain, go to the menu bar, click on edit, small window appears, click on delete, and click on the word as delete selected atoms. Just to click on top of it, and a small window, warning window appears. No problem. It says that if you are removing, it will go off. It doesn't matter. I want to remove. Just to click the word as remove, continue. Okay. Okay, I will do it. B chain has been removed now. How to find out the active site is one of the questions being asked by one of the participants. There are so many websites available. Simran, thank you so much for asking that question. There are so many sites which are available. First thing is that you have taken the PDB site. You're trying to retrieve your protein molecule from that particular site. When you're retrieving the protein molecule, make sure that there will be a literature which will be coming along with that one. So read the literature. In the literature, they will be giving very clearly exactly where is the position of the active site. So if you have the data, well and fine, you can use that one. If the data is not available from the literature, then you have certain specific sites which are available with which you are able to locate it. Okay, one is SiteHound website. I'll give you certain names of it. I think I displayed something. You can see it. Active site prediction sites are there, CAPS10, gas, and active site prediction. So many sites are available in the outsources. So you can use these sites in order to predict the active site of the protein molecule of your study. Okay. Why are not we adding polar hydrogens to the PDB format? Now, when I took the ligand molecule, I did not specify about it, but in the case of macromolecule, when you're taking the structure, you are supposed to add the polar hydrogens as well as the, you have to add in terms of other one, hydrogen bonds, what we add is in terms of Coulomb charges, what you used to say. So by using this, you have it, you can add all those things. I am just not showing it to you, but in general, when you're performing the docking process, you will be adding this hydrogen atoms also. It works offline or internet connection required. Once you downloaded the software, I think you don't require, because these are all executive files, so you may not require the internet. You only have to have your protein in the PDB format and ligand in the PDB format, which requires internet to download those compounds from the sources. So you just have to take it from there and you can start with your docking process. Okay. So that's about the few questions been asked, and this is about we have removed. Now you can say, as I remove, B chain is gone over here. I'm retaining only with the A chain molecule. Okay. Now go back again to edit. Now there may be some water molecules also may be present. So you just put delete water molecules also. It will get deleted. Okay. So now I have the structure of the protein molecule ready for me. Now we have to save the structure. So what we have done is that we have taken the protein molecule, we have removed the B chain, we have, if you want, you can add the hydrogen bonds, you can add the polar hydrogens, you can also remove the water molecule. And as I told you earlier, when you're retrieving this protein structure from the data bank, already you will be able to see that the protein's active site will be bound with an inhibitor molecule. So when I took this 4-MXO from the website, it was already bound to a drug by name called as bosutinib. So it was already associated, but that is still present in my structure, what I've taken it. So when you want to do the docking process, make sure that if there are any inhibitors attached to the active site, you're supposed to remove that one first. So what to do that one? You just go to this A chain and you just click on top of it, and you will be getting a small list of all the amino acids which are present in the A chain. So you can see all the list of amino acids. As I just scroll down, it is all coming displayed over here. Okay, so I just go down. There are so many amino acids which you are able to see it. The last one, it has been stopped near leucine 533. So now what I do is that I just down. You see now on my screen, last you're able to see something called as DB8601. So this is the drug which is already been, it's an inhibitor. Bosutinib is the name of that inhibitor, which is already been attached to the active site of the enzyme molecule. So I have to remove this one. If already a drug is attached to the active site, I cannot put another one ligand on top of it, is it not? So first I should remove it. So you just click on that one, and you see, once I do that one, you can see in the structure an yellow color is appearing, is it not? So that yellow color structure, what you have appeared is nothing but the already pre-existing ligand on top of it. Okay, so what I do that one is that go to edit, and now you go to delete, and you say it as delete the selected atom. So moment you click it, a small window appears, just say it as continue, and that yellow color one which was got selected earlier has been gone now. Okay, so this is how you are able to remove the pre-existing ligand already attached with the protein molecule. Now why this is attached? Because when I told you that when you're retrieving the protein structure from the PDB data bank center, they are already taking the protein, isolating the protein, they are crystallizing the protein molecule. So during the process of crystallization, what they do is that they add more about this inhibitor or an activator. Fivefold increase must be the concentration. So they add to it and then they do the crystallization process. So during that time, all these inhibitors will go and attach to the binding site of the molecule and then get crystallized. So the X-ray diffraction, when you are seeing it, you will be having even the ligand in the structure of the protein macromolecule. Okay, so when you want to do the docking process, make sure that you remove that one and then you do the docking step. I think B chain is still there. I know that it is still there, but I have removed it already. It is already been removed because it is not removed on the screen, but here you can see there is no B chain. Okay, so it is already been removed here, but here in the screen, it is not been removed. Okay, so no worries. It will be in the next step. I will be able to take care of it. I'll do it. No problem. Can we design drug using this docking? Yes, of course, this is a part of drug designing. So the ligand, whatever you are taking it, if it is having a pose or a conformation where a specific atom is able to interfere with the active site of the macromolecule and give you the minimum amount of the binding energy, then obviously you can consider that to be as one of a compound, as a target molecule. From there, it doesn't stop. You have to further continue with hit identification, say potential drug. It has to cross so many levels of optimization, and then only you can come into a conclusion whether it can be used as a drug molecule later. Can we dock more than one ligand in a single? It is not possible. Again, you have to read the literature. If you have the active site where it can bind only with one substrate molecule or one of the compound, then you have to use only one ligand. So it's all based on the structural pattern of the protein molecule. If it is able to bind with only one molecule, then you have to try it only with one ligand. If two are there, then again you have to see whether you can dock it with the two things. But this program is mainly we are using only for one ligand docking over here. Okay, so now whatever we have got the structure, we have to go to file and then you have to save the structure. Okay, so you can save it in the form of PDB. It says that I cannot create a PDB file because it is having one more molecule attached to it. I just say it as okay. The reason is because when you come to this current selection, you're having two compounds on my screen, is it not? One is the ligand previously, which is there. This is the green color. You are able to see that is the green color. This is the one which is already pre-existing over here, and this is the protein molecule. So when two things are there, I cannot save the structure of the protein molecule. So what I will do now is that same thing. You go to this one, select it, and go to edit, and just say it as delete the selected atom. So you can just continue. You see the ligand will disappear from the screen. So now we are left out only with the protein molecule. Now you can go to file, go to save, and write it as PDB. So this is the one, a small window appears. So you just browse it and tell, already I told you know where you want to place your information. So you can just go there and say it as PDB format and just save the protein molecule and just say okay. Okay. So now we are finished with the preparing of the ligand, preparing of the macromolecule. Now we go with the next step, the grid. So now you go to grid. Now you go to the macromolecule, then you say choose a macromolecule. Now when you choose a macromolecule, a small window appears over here and just you say select the macromolecule and just put it as select. Okay, so it just gives contains no bonded atoms. Okay, just a small window appears. Okay, now you see down in the file name, it is given as 4MXO. So just save it in the PDB QT format. Q stands for the charge, T stands for the type of the atom involved. So just you say it as save over here. So you have saved the protein. Now we'll go with the preparation of the grid box. Now again you go to grid, okay, and then you go to set map types, and there you go to the open ligand. Okay, so again a small window appears. You can just select the ligand which is in the PDB QT format. Okay, and then you say it as open. So again you can see the ligand will reappear on top of the GUI. Okay, here also you're able to see the ligand in the PDB QT format has appeared on the screen. Now again you go to grid, go to grid box. Now this step is very, very important. Now this is a grid box. The moment I select a grid box, on the middle of your GUI, you're able to find something called as a grid box appearing over here. Is it not? So it has so many different angles over here. I'm just rotating it and I'm just showing it to you. Okay. So now when you take this grid box, now what I can do is that you can see onto this window there are certain dimensions like X dimension, Y dimension and Z dimensions, and then you have the spacing and then the center grid. So now what is the purpose of this grid box generation? The main purpose of this grid box generation is that you have to arrange the grid box in the place of the active site in such a way that in the active site of the macromolecule will be present in the center of the grid box, so that when there is going to be a docking of a ligand with the macromolecules active site, there will be proper interaction between the ligand and the macromolecules active site. So that's the reason why the location of a grid box into this particular active site of the macromolecule is very, very important. Now how to get all these X dimension, Y dimension and Z dimensions? Again we have to read the literature. So wherever from the literature you have retrieved the protein structure, during the X-ray crystallography they will be telling you about even the availability of these data. If it is available, well and good, you can follow those parameters to develop a grid box around the active site of the enzyme molecule. If it is not available, very well you can go with a method called as a blind docking, one of the important facility available in this docking process, which is called as a blind docking. What do you mean by the word blind docking? Yes, you just hold on the see, this is the widgets which are available on the screen I'm showing you. So you just, here you see X dimension, it is already predefined as 40. I just hold it and I increase it. You see, when I increase it, you see the grid size is increasing, you see. So like this, you can expand in all your angles the grid box to a particular size in such a way that the whole of the enzymes goes into the grid box. That is called as blind docking. But this protein, whatever I've taken for this training purpose, is already having a defined XYZ dimensions and the XYZ centers also. So I am fixing that one for the convenience sake. So if it, you also can check it out in the literature and you can do it. If it is not available, then you can do simply the blind docking. Many of them prefer to do even the blind docking also. So as I told, blind docking means what? You are going to make sure that the whole of your active site and the protein component is present within the grid box. Okay, so now similarly here also X centers, I am just changing the parameters. I will share with you this manual. You can download this protein and you can download this ligand from the centers, what I'm going to share with you later, and you can try to do them by yourself also, because manual is self-explanatory. So you can see the manual and you can do it. You can experiment it once, later depending on your convenience you can check it out and try it. So these are the, see, I have defined the parameters. I will show you the binding energy at the last. Receptor flexibility. When you take two different ligands, comparing, you check out for the binding minimum energy. Binding energy is a matter over here that will come to the last one, so that you can finalize out of 2 or 3 or 4 drugs, ligands, whatever you're choosing, whichever ligand with a particular pose or a particular conformation giving you the minimum binding energy, that will be your future target molecule, which you can use for the process. There is no huge difference between them. They are only the tools which are available. Autodock Veena is this like something like pre-existing Autodock. It is trying to repair it, or which one is? Both are specific and fast. You don't have any issues with them. Any of the softwares you can use, Autodock or Autodock Veena. Both of them you can use. They are good software tools. Okay, ma'am. I'll continue with the next step. That is already we have defined where is your, see now as I move, you are able to see the grid box. You can see some of the loops and the chains which are present here inside the box, some are present outside. Inside we are not able to view it, but you are able to see the grid box developed over here. Okay, so just go to file and say it as close saving current. So it will get saved up. Okay, now once it is saved up, you have to save it in a file extension file name. That is very important. So again you go to grid, go to output, and then you go to the save GPF. GPF stands for grid parameter file. Just click on top of it and you save it in the file name as grid.gpf. Okay, and just you save it. Okay, so once this is over, now we'll go with the next parameter called as a docking procedure. So go to docking, go to macromolecule and set the rigid file name. Okay, so again you will, a small window appears. You select the macromolecule in the PDB Qt format and say it as open. Then again you go back to your docking. These are the steps which you have to follow for running the docking steps. So go to open and it will ask where is your macromolecule, your ligand. Already I have ligand is in the PDB Qt format, and just say it as open. Okay, so a small window appears. You can just click it as accept. You can just go through it. It says active torsion, the ligand is 7 and few other details. You can just say it as accepted. Okay, so now we'll go to the next step, docking. Go to search parameters and go to genetic algorithms. Now a small window will appear for you. It is asking how many algorithms, genetic algorithms you have to run. That is 10. So it is like a metric system, this genetic algorithm. So you have certain population size. It is actually trying to interpret the data in terms of genetics. Like in the case of fitness, we say that you have the Darwin's theory of evolution. There is a fittest one, is a survival. So out of so many ligands with so many posts and conformation, which is the best one fitting into the active site. So that is what is trying to do with the help of this genetic algorithm. So by default, you just say it as accept. You have to 10 as taken, you can take it 100, or it's up to you. You can just accept it. Okay. Now you go again to docking. Then go to docking parameters, and then you just a small window appears and you just say it as accepted. Okay, so the next step is going to be the docking output. So go to docking, go to the docking, go to docking, go to output. Again, a small window appears. Lamarckian GA stands for generic algorithm 4.2. And now it asks, how do you want to save the information? Whatever is a feeding I've given to you, you just say it as doc.dpfc. Please, participants, you note down this file name. GPF I'm putting, DPF I'm putting. So this is doc parameter files. This is the files which will be recognized later when you're running the Autodock and the AutoGrid process. So you have to be very careful with that one. Okay. So now we'll go on with the next step, that is called as a running the Autodock process. Okay. So now what we'll do is you go to run, and then you have to select this run AutoGrid step. Okay, so a small window appears over here. It will ask you path name and the file name. So you have to define where is the path name and the file name. So you just go to click browse, and you see all the files has been saved over here. Whatever I did now in the same folder, earlier folder was having only 4MXO and this and few other documents. Now you see there are so many other documents placed over here. So you have to be very careful the path what you give, where your documents are present. Okay, so since we are doing the AutoGrid, you just select the AutoGrid over here. This is an executive file from Autodock suite. You can download it and take it. Okay, so just you click on this AutoGrid 4 and say it as open. Okay, now this is the path name you are defined. Now come to the file name. So just click on top of it. Again, it will ask you some data. So already when I clicked that one, that is called as the parameter file name. I have deposited the parameter file name in the grid GPF format. So it is in the GPF format. Just click that one and say it as open. Okay, so the moment I click on the file name, the log file name will appear directly on the screen, and just click on the words launch. A small window will appear and disappear. So it is doing all the calculations. Okay, Now we'll go to the next step. That is, you are going to take run. First auto grid informations, you have fed the data. Now it is going to be the run Autodock step. So just click on Autodock. Again, the small window appears like this. So you have to define the path name, file name, and the log file name. So first, already it is Autodock 4 is there, but don't go, just leave it off. You just have to define it again one more time where it is present. So better that you go back to your folder, select that Autodock 4 executive file and click it on open. The next one is a file name. Already it is existing, but you go to your file name and see already we have generated at the doc DPF file. You just click on top of it and give it as open. So even the file name has been generated. So you just put on the word as launch. So it is doing the auto docking and the auto grid working process has been started now. Okay, now the next step is analyze step. Okay, I'll just show you the steps. So go to analyze, go to dockings, and just you say it as open. So you already there is a file, DLG file, which I have generated earlier. So because I have to complete the process within a short span of time, so that's why the DLG file, that is doc logging file, has been already been prepared. So I just click on top of it and say it as open. So I already prepared that one. The prepared one, I'm using it for today's purpose. Okay, so now it shows you that new name ligand 19.3. Just click on OK. Okay, when I do that one, it says that only there is a red one docked confirmation from the doc DLG file. Just click it as OK. That's it. Okay, now you see this is the ligand which is been added up over here. Now again go to analyze, go to macromolecule and click on the word open. So now again the protein has come on top of our screen. So now you can select the protein. Already it has been selected. So what I will do is that the previous one, what is existing, I will try to deselect it. 1 minute. I don't know, something is happening to my screen. What is this? Okay, I'm gone to the dashboard. I'm just selecting the previous existing one. I'm clicking delete and I'll delete the selected items. Just say continue. Okay, so I'm left out only with the one protein what we are going to use now. Okay, so now I will click this one protein and make it to show you in a format that is a ribbon format, so that you'll be able to see it. Okay, somebody, okay, you are able to see the protein now. Okay, so I'm just rotating it. Somebody said, ma'am, you're having both the chains together. So it didn't get removed there, but when I was saving it, it was saving only with the A chain. Okay, no worries. There will be some hitches. We have to just learn how to understand them. Okay, so earlier it was there. Now you see, when I saved it, it was saved with only the A chain. Okay, so now this is over now. So what we will do is that we will go to the next step, analyze, and go to confirmations. I hope you are seeing the screen. You're going to click it as play ranked by energy. The moment I do this one, there is a small window which will appear on my screen. So what we can do is that there are so many features. I will just click on this particular symbol to tell you something, because it is 11:45. The next session should start. So I will just show you with the result and wind it off so that I can give space for the next lecture. Okay, so now a small window like this will appear on my screen. I hope you are able to see it. So just click on the word as show info. So now you are able to see some information available. See, this is the binding energy. Okay, so minus 6.78. It's minus 6.78. So the ligand, whatever I have taken, one of its atom was able to interact, okay, with the macromolecule, and when it is interacting, the energy, binding energy, has arrived to a minus 6.78. I have tried only with one ligand, with only one particular pose or a confirmation, my dear participants, that I'll give you clarity. It's only one ligand with one pose or a confirmation. I'm showing it to you today. So that is why I'm showing the energy minimum as minus 6.78. Okay. Now this is one thing, and you can see various other inputs also which are available over here. Next I will show you the hydrogen bonds building over here. So just click on top of it, a small window appears. It is saying show the distance. So let me show you where is that hydrogen bond and the ligand. Okay, so I don't know whether you're able to see it. Maybe I will zoom it out for you so that you can see the structure. I hope you are able to see something in the I will, I don't know whether you're able to see it. I will show you in different format also. As I rotate, you are able to see in the center something like this number. Okay, so that is showing you the distance of the hydrogen bond which has formed over here. Okay, so I'll just, you are able to see participants. This is the last part, very important part. Whatever we are doing, execution of each and every file is, steps are very important. Then only you can come into this particular step. Okay, so now what I will do is that I will make it more comfortable for you in the viewing. So I'll go to this ligand, that is the third one which has been taken up by the GUI. I will just select it. So now we are able to see, I think. So you see the ligand now, it is present in the enzyme. It has docked over there, and one of its atom has been, I will show it in another format also. So you can see now, you can be able to see. So you see now, I'm trying to rotate it. Maybe I'll close off all these things so that you can view properly the ligand and the protein interaction. I will show you again the another. You are able to see, my dear participants. This is the last important section of it.
You're able to.
Okay, ma'am. Okay, ma'am. I'm almost finishing it off with one or two lines. I'll just finish it off now. So this is the ligand. You can see earlier when we took the ligand in one format in the first stage PDB format. It was in one particular shape or orientation. Now we are able to see the conformation of the ligand as changed. Okay. So for convenience, one more time I will show you the bonding also, hydrogen bond. So I am showing the distance. Yeah, you are able to see it, something like blue color. So you are able to see here, you are able, that blue color one is a bonding. That's it, ma'am. I am done with it. So thank you, participants. Whatever I have learned
Take some
from,
questions.
sure, ma'am.
Shall I
So,
invite the participants unmute themselves?
sure, ma'am.
Ask questions.
Okay, ma'am.
So I'd like to make a small request to all the participants, and Mrs. Vanita, she is a faculty member, been teaching the subject from past 3-4 years, and whatever the knowledge she has gained, she is able to give it back to the audience. So expected to her level of understanding. Maybe some of you are there in a much more advanced level, maybe at your research level, or maybe whatever she has made and a lot of effort to make the people who are the beginners and who are at the stage of understanding what is molecular docking, because many of us, we don't know what is molecular docking. So the intention of this workshop is to provide awareness, and it is a part of the curriculum of some of the syllabus. So our intention is to make the participants to understand the basic concepts of Chemsketch and the molecular docking, and use our resources which are available in the department to spread the knowledge what we have to the participants. So with this intention, I am making the participants able to unmute themselves. So you please ask if there are relevant questions.
Somebody is asking me while I'm doing blind docking how to check out whether it is good or whatever it is. See, you have to experiment it, ma'am.
I have allowed the participants to unmute themselves if anybody is willing to ask orally.
Yes, ma'am. Thank you, ma'am. It was really a wonderful talk.
Thank
My
you so
question is,
much.
ma'am, without using Segment, ma'am, because Autodock tools generally use Linux and Python. So, ma'am,
Yeah, yeah,
without
yeah, yeah.
using Segment terminal for the Linux based, how we can use the docking procedure without
See,
using.
that is where I told you doc DLG file which I generated. I told you is it not which I already previously have done it. So again, you will require the program in order to convert the RMSD. That is a root mean square values. You have to take all those values and convert and prepare the doc DLG files, and then you have to do it. It's again a long process. You require more software. See, we have a time constraint of 2 hours. I have to make you people to know that this is one tool which is existing, and you can experiment it later to understand more about it. Okay, so you have to experiment and you can learn it. So
Yes,
there are
yes,
some
ma'am.
tools available, so you can do it. You
Yes,
can do
ma'am.
it in that
Yes,
format
ma'am.
also.
Ma'am, my last question is, ma'am, here
Yes.
the protein and the ligand we have, it is known to us the structure.
Yes.
Suppose we have identified a protein whose structure is not known, and the ligand protein interaction is not known. Then how we can?
I gave 4 strategies in the beginning. That's why I
Yes.
started with the 4 strategies. Okay, so depending on the
Okay.
available data, you can choose which method you want to follow for
So,
doing
ma'am,
the
we
study.
can talking to a approximate
No,
level.
yeah, you can do it, but you have to use some other software tools in order to do when you don't have a known data with you.
Thank you, ma'am.
Okay,
It
so
was
yeah,
really a very good talk.
thank you so much.
Hello, ma'am. I was
Yes,
having a doubt that whenever we do docking,
yeah.
we after that we visualize it in PyMOS. So that time we are only seeing the polar contacts which the drug and the protein is having. Is there any way we can visualize all the contact that a protein has with ligand? Because here also only hydrogen bonds were visualized.
Actually, when binding energy is being taken, we talk in terms of hydrogen bond, which is very important. So that you have to see, you have to finalize your ligand, and you have to specify which atom. I didn't actually show you the PDB coordinates. That is a final output also. You will be getting the PDB coordinates in the notepad. It will be coming out. You have to understand the PDB coordinates over there. For each and every atom, they'll be giving the XYZ coordinates, and also the details will be there in that one. We have to understand. Then you can have some idea about the atoms and its interactions with the ligand and the macromolecule. So that again requires so much of understanding about all those things.
So the most important interactions there are, those are the polar contacts. Other ones are not that important in
It's
terms
not
of
like
binding energy
no
and
no no
everything.
no 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 not like that. See, that is what you have to see the PDB coordinate files. Ma, you have even non-covalent interactions also, like hydrophobic, van der Waals,
Yes.
electrostatic. So whatever energies are being given, no, they will be having one way of calculating it and converting it into the energy which is required for that one. That and all I didn't show it to you, because in the grid parameter file and the DPP, that is a talking parameter file, we have done, is it not? So in all those things, and even in the DLG file, you have all these data available. You have to first understand that one, then only you can understand the atoms and the atom which is interacting with the macromolecule also. Okay, so we have to understand. See, it's everybody can run the docking process very easily, ma, because you have the tutorials available even on the website also. You can run it very smoothly, and even the manual begin, you can run it. But how do you interpret your result is the understanding about the subject. So you have to take all those results and try to check it out individually. What are the coordinates and what are the atoms, what is they will give you very clearly. There are 7 torsional angles over here is given, some 7 angles are given, so you have to understand that one and then come into certain conclusions. Okay, so it requires lot amount of theoretical, practical, so many. This is a biology with the computer technology. Okay, so we have to have more amount of biological informations, knowledge about the subject also. Thank you for asking me. I think
Thank you,
something
ma'am.
whatever I know. Ma'am,
Yeah,
sir
any
has
more
come, ma'am.
questions from the audience, or we will break for 10 minutes and the next session starts with Mr. V. Ramesh.
Sir has joined, ma'am.
Yeah, he has already joined, but we'll be back after 10 minutes.
Okay, ma'am. Participants, thank you patiently listening to me. So this is my first time I'm giving it. So if I have made any mistakes, please excuse me for the first time, and thank you for patiently listening to me also. Okay,
Sunita,
thank you so much. Yes, ma'am.
you please stop the recording and we will start again because the file size is becoming too much to upload.
Okay, ma'am. I will
Please
stop
stop.
recording.
Pause